Let us now talk about the plasma membrane. The plasma membrane was named by Nacelli. It is the limiting boundary of each cell which separates the cytoplasm from its surrounding. It is present both in plant cells as well as in animal cells. It is the outermost covering of a cell in the case of animals, and it lies below the cell wall in case of plants. The cell membrane is also called as plasma lemma or the plasma membrane. The names it has is the cell membrane, or it may be called the plasma membrane, or it may be called the plasma lemma. The model which is most accepted as a model of the plasma membrane was given by Singer and Nicholson, and they gave the fluid mosaic model for the plasma membrane. According to them, plasma membrane consists of a protein layer sandwiched between two layers of lipids. And the plasma membrane is a quasi-fluid state. And it is 75 angstrom thick. According to Singer and Nicholson and the fluid mosaic model which they gave, the plasma membrane consists of a protein layer sandwiched between a bilayer of lipids, and it is in the quasi-fluid state. It is about 75 angstrom thick. The plasma membrane is selectively permeable in nature. That means it allows the entry of certain necessary molecules and which are necessary for the cell, and it restricts the entry of substances which are not required by the cell. If we want to understand the diagram of the cell membrane or what it is made up of, we can understand it somewhat like this. It is made up of a bilayer of lipids. It's a bilayer. So this is a bilayer of the lipids. And protein embedded inside. Some are the peripheral proteins. These may be the peripheral proteins. Some are between the lipids. Sometimes the proteins form a structure which allows the entry of certain molecules easily. So these are the phospholipids. This is the tail of the lipid, this is the head. The head is hydrophilic and the tail is hydrophobic. These are the proteins, peripheral proteins. This is the pore which is formed by integral proteins, and it allows the entry of certain molecules. This is the glycolipids, sugar attached to the lipids, and this may be the glycoproteins. These are the integral proteins. There is cholesterol also. And this we can say is the phospholipid bilayer. So from the diagram we can understand that the plasma membrane consists of a phospholipid bilayer in which proteins are sandwiched. There may be peripheral proteins and integral proteins. Sometimes integral proteins form a pore through which it facilitates the entry of certain molecules. These are the glycolipid and these are the glycoproteins. So diagrammatically, this is the fluid mosaic model of the plasma membrane. Let us now talk about the functions of the plasma membrane. The first and the foremost function of the plasma membrane is holding the cell contents. It holds the cell contents, the things that are inside the cell. It gives protection to the cell. It gives individuality to the cell. That means two cells can be differentiated by its presence. It gives individuality to cell. It holds the cell organelles in their place and delimits the cell organelle. So they have a definite home. It helps in cell recognition. With the help of glycolipids and glycoproteins, it is able to differentiate similar cells from dissimilar cells. With the help of glycolipids and glycoproteins, it can differentiate self from foreign. cell recognition is useful for tissue formation and defense against microbes. The sixth function, the cell membrane possesses antigens which determines blood grouping, acceptance or rejections of a transplant, etc. The cell membrane possesses antigens which determine blood grouping, acceptance or rejections of a transplant, etc. The next function of the cell membrane is cell junctions. They help in keeping animal cells together. They act as cell junctions and help in keeping animal cells together. The next function is microvilli. There are structures or invaginations of the membrane which are specialized for increasing absorption of nutrition. The next and the most important function of the cell membrane is transport of molecules. As we all know, the plasma membrane is selectively permeable. That means it selects the substances which enter the cell and exit the cell. Some substances enter and some do not. When we are talking about transport of molecules, we should know how transport of molecules takes place across the plasma membrane. Transportation of molecules across the plasma membrane can be done in the following ways. One, by simple diffusion. This means the movement of ions or molecules from a higher concentration to lower concentration. The examples of this can be the movement of CO2 and O2 across the membrane. So CO2 and O2 diffuse through the membrane. The second way in which the transport of molecules can occur is osmosis, and it is related to fluids. Takes place only in liquids. Osmosis is the movement of solvent or water from lower concentration of solution to higher concentration of solution. Osmosis can also be called as diffusion of solvents, and it takes place through a semi-permeable membrane. We can also define osmosis as the movement of solvent or water from its higher concentration to its lower concentration. The movement of solvent into the cell is called endoosmosis. Entry of solvent inside the cell. And the movement of solvent outside the cell is called exosmosis. Both diffusion and osmosis do not require energy, so both of them are called as passive transports. Passive transport is a transport which does not require energy. The second way in which molecules can move through a plasma membrane is with the help of special proteins called transport proteins or permeases. It is a mediated transport. With the help of certain substances or certain proteins, molecules move across the plasma membrane. Permeases form a small passageway through the membrane, enabling the solute molecule to cross the phospholipid bilayer. Permeases are usually specific. That means they recognize and transport only a limited group of chemical substances or perhaps even a single substance. In facilitated transport or diffusion, the permease assists a molecule to diffuse through the membrane that it cannot otherwise penetrate. That means it forms a smooth entry or a passage for the entry of the substance. When we are talking about the transport of molecules across the plasma membrane, it is necessary for us to discuss the types of solutions which are based on concentration. Depending on the concentration, a solution can be isotonic solution. In the isotonic solution, the concentration of the solution outside the cell is equal to the concentration of solution inside the cell. The second type of solution depending on concentration is hypertonic, in which concentration of solution outside cell is more than that inside the cell. The third type of solution depending on the concentration of the solution can be the hypotonic solution. Hypotonic solution is a solution when the concentration of the solution outside the cell is less than that of the cytoplasm of the cell. Is less than the cytoplasm of cell, or inside is less than the concentration of solution inside the cell. Depending on these solutions, transport across the membrane is different. In a hypertonic solution, the cell loses water and becomes plasmolized. In a hypotonic solution, the cell swells up and bursts because the movement of water is inside the cell from its higher concentration to its lower concentration. So, in the case of hypertonic solution, the cell loses water and is plasmolized. In the case of hypotonic, solution, the cell swells up and bursts because water enters inside the cell. In an isotonic solution, the concentration of the solution outside the cell is equal to the cytoplasm of cell, and so there is no movement of the solvent. So these were the three types of solution based on concentration of the solution.